Sickle cell anemia. First, let us go through the key fact. This disease, it is a point mutation. What is a point mutation? This is a substitution mutation. See here, adenine is replaced by what? Uracil. It can be GTG also. That is, if DNA it is GTG and mRNA it is GUG. Okay, so substitution mutation, and that is called as point mutation. And then it is a beta globin gene mutation, and then the mutation at 6th amino acid. Since GAG becomes GUG, glutamic acid will be replaced by what? Valine. And then it is an example for autosomal recessive inheritance. If HBA it is normal, and HBA-HBS it is carrier, and HBS-HBS it is affected. First question is sickle cell anemia. It is caused by what we discussed? Point mutation. And then in sickle cell anemia, glutamic acid is replaced by what? Valine. And the next question, the mutation in sickle cell anemia occurs in gene coding for what? Beta globin chain. And then sickle cell anemia is inherited as autosomal recessive trait. Then which codon change occurs in sickle cell anemia? That is GAG becomes GTG. And if it is a DNA, it is GTG, and mRNA codon, it can be GUG. Sickle cell anemia of RBC mainly occurs under low oxygen concentration. The abnormal hemoglobin in sickle cell anemia is HBS. And then which statement is correct? Heterozygous individuals are completely normal. The heterozygous individuals are actually carriers. Okay, we discussed HBS and HBA. HBS and HBA. They are carriers. And then homozygous individuals suffer from severe disease. Yes, homozygous individuals, that is HBS and HBS. And the next question, 9th question, sickle shaped RBCs mainly lead to what? Anemic condition due to hemolysis. Next question is, a man and woman both are carriers. So the genotype is HBA-HBS, right? And then you have to find out the probability of sickle cell anemic children. Okay. And since both are carriers, the parental genotype will be what? Heterozygous, HBA-HBS. So both produces two types of alleles. Father produces HBA-HBS and the mother also produces HBA-HBS. Why? Because both are carriers. Okay. Then Punnett square, the alleles are HBA-HBS, and so the genotypes will be, that is normal, right? And HBA-HBS, this is carrier, and here HBA-HBS, this is also what? Carrier, and this is what? Deceased condition. Clear? HBS, that is what? Deceased. And so the normal is 1, and then the carrier is 2, and then the affected or deceased is 1. So the ratio is 1:2:1. 1 normal is to 2 carrier is to 1 affected. Okay. And our question is find out the probability. That is probability of sickle cell anemic children. So that is 1 out of 4. So 1 by 4, that is 25%. So our answer is 25%. Okay. And next question is, normal homozygous individual HBA marries the deceased person, that is sickle cell patient HBS. And what will be the genotype of all offsprings? Same method. The parents are, one is normal and the other one is deceased or affected, that is the patient, HBS. So the normal parent produces, since HBA, it produces only single type of allele, that is HBA, and the patient produces what? HBS. And this is the Punnett square, and the condition is HBA-HBS, which are carriers. Okay, so all the individuals will be what? Carriers. So all children will be HBA-HBS, that is 100% what? Carriers. Okay, so the answer is option B, HBA-HBS, that is all individuals are carriers. Next question, a carrier marries a normal homozygous person, what percentage of children are expected to be carriers? So the first step, the genotype of parents. One is carrier, that is HBA, HBS, and the other is normal, HBA, HBA. Okay. And they produces the gametes. The carrier produces, since heterozygous, HBA, HBS, and the normal produces what? HBA. And then the Punnett square. This is the genotype. HBA, HBA, that is the normal person. And then HBA, HBS, they are the carriers. Okay. So the percentage of, we have to find out the percentage of carriers, right? So normal is 1 and the carrier is 1. That is, the ratio is 1:1, and total 2 individuals, and 1 is what? Carrier. Okay. So 1/2, that is what? 50%. That is the answer. So the probability of carrier children is half into 100, that is 50%. So the answer is B. And the next question is, sickle cell patient, that is HBS marries a carrier HBA, probability of sickle cell anemic children. So the first step is the parental genotype. The parent is, one is what? Normal. So one is deceased, right? Patient or deceased condition. One is patient, that is HBS, and the other is carrier HBA, HBS. And so the gametes from patient HBS and from carrier, that is HBA, HBS. So this is the Punnett square. HBA, HBS, this is what? Carrier. And HBS, HBS, this is the patient or deceased condition. Okay. Here also the ratio is 1:1 and the percentage of carrier is 50%, right? The probability of the patient or affected person, half into 100, that is 50%. So the answer is 50%.